Okay, today will be the lecture 4. And in previous lecture, we know that the several sites in the N-terminal is critical for forming dimer, and some sites are important, like in a constructional site. But I'm wondering why it didn't mention where is the Schiffer base of the alpha trans retina binding. Before I know that this 11 says transretinal binding to human rhodopsin is at the transmembrane domain 7. So until I read this chapter 4, I know that it's the same. It's quite conserved around the whole world of rhodopsin. That's the rhodopsin is also in channel rhodopsin and other like chloride pump, the bacterial rhodopsin, her rhodopsin, although the binding ligand is all transretinal, but you are still binding to the uh, uh, lysine residue of the transmembrane number 7. Today, I'm going to move on to the lecture 4, and it's a functional mechanism of chloride pump, rhodopsin NC conversion to proton pump. Then let me try. So, and today the outline we're firstly, and we're going to talking about the phylogenetics relationship of different proton pump or chloride pump. And the next, and we're and keep focused on the tertiary structure of the pump and the photocycle of a chloride pump. In the whole rhodopsin families, for like the heterodopsin, the chloride pump, or bacteriodopsin, the proton pump, and it will cause the hyperpolarization of the cells or the microbial creatures that expressed these proteins. In long terms before, people think that a pump may be not so efficient as the channels because pumps require higher protein expression level and stronger light stimulation, because each photocycles, they can only like pumps one time of those cations or ions that are binding inside into this pump, but the channel were different. When it's open, it just like on flowing pathway for those cations. However, due to like maybe like some pumps that there's like a conformation change that between the two status within one photocycle. So the pumps can also like successfully hyperpolarize like neurons in the like experimental approaches. So first, let's take a look at the physiological relationship between several pumps. And in this phylogenetic tree, we can see that actually the proton pump is from the evolution, its relationship is quite close to the chloride pump. So the bacterial rhodopsin is quite close to the heterodopsin for the NP. And there's other groups of chloride pump like NM3R, and this is called NTQ. That's based on like this, the critical part, like a motif, NTQ motif, and we'll discuss later. And this group are coming from the deep sea, and the TSA chloride pump are coming from like a forest, those bacteria or microbians. So NPHR is a TSA motif, and the bacterial rhodopsin is a proton pump, and it's a DTD motif. And other like XR or GR is a DTE motif. It's also a proton pump. Of course, there's other sensory rhodopsins. So here, this is the table 1 that lists all those pumps we'll be talking about discovered today. So for the MRHR or SYHR, and it induced influx of chloride or other halide ions like a bar, like iodine, like sulfate. And in the next slides, and I'm going to call this MRHR, Mr. HR, and we can see that like for Mr. HR, it will induce some halide ions that pump into the intracellular part. Unlike other is YHR, this group is quite large, like halide, sulfate, nitrate, iodine, bar can all be like pumped into the cell. Then what's the functional of this work? So, and the author of this chapter that provided several hypotheses that maybe it's important for ATP production in some bacteria, but in other bacteria it also have this function, but it's not really important for the energy produced because it's some other like microbial, it will have a function like photosynthesized like plant. So another possibility is that it can maintain the cellular osmotic balance because between the different conditions, this microbial might change the shape. It becomes larger or it will shrink. So all this need to require that a balance of the cellular osmotic. So it's much easier for the cations because the cations there's other like channels or pumps that can freely like build the smaller sites. But however, for the negative charge of the intracellular part, it's like a very large like proteins or lipid or large molecules. That's not so easier like can go inside out of the plasma membrane. So this could be important that to maintain is serum osmotic balance and to like pumping in these negative charged ions. And another possibility is that maybe it can help the cell's survival in non-aqueous environment, like increase the salt concentration, and it may help that this like preserve the cell's viability to survive like some very harsh environment that without water. Then let's move on to see the structures. In previous lecture, like talking about some important site like the 3-cysteine residue that is critical for dimer formation for only channel rhodopsin, and the glutamate residue that's important for the ion selectivity or permeability and in the constructional site. And also, it's mentioned in the beginning of this lecture, that's the transmembrane 7, that's this lysine residue is important for the ligand and all translating binding site to form the Schiffer base. That's on the lysine. So it's said that in the following lecture, we're talking about some very important site that is important for the function of those proton pump or chloride pump. And we'll also see that these sites, when it put together side by side, and we know that this side are like the same side if it's a positive charge or negative charge, or it's a hydrophobic residue that will dramatically change the function of this pump. So in this lecture, we're going to talking about the bacterial rhodopsin, the rhodopsin, and an MR3 that's and DTD, I think. I'm not sure. Okay, so the bacterial rhodopsin is from the archer rhodopsin and also rhodopsin, and the NMR3 is eubacterial rhodopsin. So bacterial rhodopsin is a proton pump that pump out the proton, and the rhodopsin and MRR3 as pump in the chloride. Pumping both can pump in the chloride. And the most sensitive wavelength for bacterial rhodopsin is around 568. And it's a 575 for rhodopsin, and it's a 534 for MRR3 chloride pump. And now let's move on to see the tertiary structure of those pumps. And we can see here that like this is a so-called the DTD motif, that this residue is quite important for the function of the bacterial rhodopsin. It's also we can see that all this amino acid is quite important for the function, like the ion selectivity, ion permeability, and the photocycle kinetics of these rhodopsins. And from this image, we can see that it's not only the selective ions that is important for rose pump. The water molecules were also involved in this hydrogen hydrogen bond network, and it helps maintain the structure, and it helps like binding those ions that is being captured by this pump. And there's some like very detailed difference between different chloride pump, and these were all determine the photocycle or kinetics of the pump. So firstly, let's take a look at the bacterial rhodopsin. The bacterial rhodopsin will pump the proton that pumping out of the cell. So firstly, the proton will come into this direction, then binding to the aspartate 96 amino acid, and then it functions as like a proton donor to the protonized Schiffer base. And when it's light actived being captured by the retinol, then this proton will be like translocated to the sp85. Then through this like this several translocation that change from this residue to Schiffer base, then to another residue, and all this is how the proton that be pumping out from the bacterial rhodopsin. And this residue is like we can find like the similar positions at rhodopsin and NMR3 rhodopsin. It's a chloride pump. That's the serine 126 or the serine 74 on the Mr. HR, or as paragen 98 in MR3. So these are all like correlated to each other. And now let's move on to see the network, the hydrogen bonding network. And we can see that chloride ions are surrounded by intensive hydrogen bond network, like in rhodopsin and NMR3. And this hydrogen bond network is also required the water molecule. And this different interaction between chloride and protonized Schiffer base might be a reason for different photoreaction. So we can see that the hydrogen bond network is slightly different between the rhodopsin and MR3. So it's like the hydrogen bond network here is like a huge network, and here like this network is quite separate. So it's like these two water molecules like forms a hydrogen bond network, forms like to stabilize the structure. And this part, in chloride pumped to link to another part. But in rhodopsin, all these three parts are linked together. So this will all determine the characteristic of this different the chloride pump. And now let's move on to take a look at the photoreaction cycle. So this diagram shows the photoreaction cycle, and the inner cycle is more focused on the pumped function. To see that like this determined by how the conformation change, how the ion like its open state or its closed state or its decayed at inactive state, something like that. And the outer is more focused on the light response to see that at this state that these channels are more sensitive to certain kinds of light wavelengths. Let's first take a look at the MPHR. And MPHR at the resting state we labeled at the P0, and the other different intermediate status is labeled like a P1, P2, P3, P4, P5 in other chloride pump. And we need to mention that in this, after like activate, then switched from the resting state to K to L, L1, L2, then it goes into a crescent equilibrium state. And at this state, actually, like this pumps it happens almost at the same time. And depending on the membrane potential or the concentration of the chloride, this is two state can switched like freely between these two states. And N state is a blue shifted N state, and O state is a red shifted status. And when the chloride is free, intracellular is no chloride, so these cells this pump will be like prone to stay in O to stay in O state. But since this O state and state happens together, and like a like exists, it can switch between these two states. Then which status is more close to the HR prime status, and which one is the upstream step? So here in this lecture, we're going to introduce a very interesting and a very clever method, and that is developed and published by this articles on this review chapter's author Kikukawa that published in 2015. So here they use a very clever method that for the preparation of this channel, they add the carotenoid bacteriorubenarins. This kind of a molecule or like a lipid, and also have some light absorbance property that mix together into this preparation, and this bacterioruberin will only like have the wavelength response and when it binding to the O state, but rather than not at the N state. And this image is from the original article. That this bacterioruberins will only have wavelength changed when the pumps are in O state or HR prime state, but not in N state. So here we know that since this there's like the functional change or like a reaction change of the bacterioruberins in the O state and HR prime state. So here we know that like there's a more closed between the O state and HR prime state. From the functional or it's from the response because their response are more close because it's only like have some change in this two state. So HR prime is more close to the O state rather than in the N state. So this experimental approach can discover the detailed like steps that is first is the N state, then to the O state, and there's a translocation of the chloride from N state to the O state. And after binding the chloride, then it will change to the O state to further decay to the HR prime state. So here we're talking about an experiment, a research method to make this discovery. I think it's quite smart. And this is how like scientists to make discoveries is pretty cool. And now let's move on to see that in the photocycles, the sugar base will remove the water molecule. So when the water molecule is being removed, so the hydrogen bond network will be damaged and will be destroyed. So and the binding of chloride was not like so stable. So it caused this force, this attractive force is lower, is reduced, and it will cause the chloride like to move to other place. And in previous, we're talking about the photocycle of the NPHR, and it's quite similar that happened in Mr. HR or an MR3, but the two state, the N state and O state in Mr. HR is more close and hard to separate these two states in Mr. HR, and the Mr. HR structure is not directly discovered yet. So there's still some research gaps here that need to be further studied. And for the NMR3, there's no blue shift state. There's only two red shift state, O1 and O2. So after we know this, the function of this amino acid residue, and in the next step to we can do some engineering work and also to provide more evidence to make sure that confirm that we know this residue's real function. So that's to make the point mutation to change the whole pump's functional characteristic. So here is that in the Mr. HR, a single mutation that the serine 74, and in previous slides that we talking about that this style is quite similar to the asparagus 85. So only make these single mutations and to change to asparagus 70 should be negative charged amino acid residue can change the whole chloride pump change to a proton pump. It's like it has a very weak positive charge when it changes to a negative charge. Then this two negative charge cannot form like a stable so before they were like negative, positive, they will have a attractive, but if this side becomes a negative charge, then the only attractive will be a positive charged cations like a proton. So one single mutations can change the whole channel to different function, that from the chloride pump to the proton pump. So here is the experimental result to show that this one single mutation will cause the whole light absorbance property changed between Y type Mr. HR, mutant Mr. HR, and this mutant Mr. HR, it changed after it changed to proton pump, and this functional is also being sensitive to the pH. So we know that like proton pump that's what close linked to the pH status of the intracellular or extracellular. So this provides another solid evidence to know that this channel is being more sensitive and related to the concentration of the proton. But this channel need further optimization because that from the P3 status change to the P4 status, and this takes a very long time. It's 4.7 milliseconds, and that's the rate limiting process in this channel's function. So now let's summarize those very important and scientific steps that in 1995, Sasaki group, one single mutation changed this acid A less aspartame 85 to serine 85. Then one changed the bacterial rhodopsin from a proton pump to chloride pump. And in 2019, in MRHR, then also the same site changes serine to aspartame that changed the chloride pump to a proton pump. And we're gonna see that it's like a switching back, like SP to serine and the proton pump, chloride pump. Then serine to SP, then the chloride pump to proton pump. But this step is only successful in Mr. rhodopsin. It's not in NP rhodopsin. It's not successful. So in another like a background Y type rhodopsin, and this step is became successful. Then let's go back to take a look at the whole tertiary structure of this rhodopsin. So beautiful and so nice, and some site that plays a very important role in the whole function of these proteins. So thank you and see you next time.